Welcome back. Today's lecture is going to be on microbial mechanisms of pathogenicity. What does that mean? It means how do microorganisms cause disease? We know how our immune system functions to protect us against disease. Now we're going to see how can these microorganisms evade our immune system? Okay? So let's go ahead and get started with today's lecture. A quick overview of pathogenicity. Usually, the way that microorganisms enter our body is the same way that they will exit. So usually the portal of entry or the portal of exit is the same as the portal of entry. So the way that you became infected is usually how you proceed to infect others. Okay? So how do microorganisms usually enter our body? Various portals of entry include the mucous membrane. The mucous membrane includes the respiratory tract, the gastrointestinal tract, genitourinary, and conjunctiva, the eyes. There is also the skin and the parenteral route. Now, what is going to depend as far as if you get an infection and disease is the number of invading microorganisms. What's the viral load? What's the bacterial load? How many microorganisms entered your system, right? So the number of microorganisms that enter your system may then adhere and possibly cause infection. Now, how do they do this? Penetration. How are these microorganisms able to evade our host defenses? We already went over the innate immune system. We went over the adaptive immune system, and we know how effective they are, right, at combating microorganisms. So you may be wondering, they're so effective, how do we ever even get sick then, right? Well, microorganisms can have various things that protect them from our own immune system, including capsules. We know what the function of capsules are. We've talked about that since the beginning of the semester. Capsules protects the microorganism from being phagocytosed, right? Cell wall components, we'll talk something about that a little more, enzymes, antigenic variation, etc. And then these organisms can cause damage to the host. These are the various ways that they can cause damage. So we're going to focus on some of these toxins that some microorganisms can produce. And then once they cause their infection and they damage the host, they can proceed to exit your body. Generally, the same way that you got infected is the way that you will proceed to transmit the disease to others. So how are these organisms able to invade our body? For one thing is that they can penetrate the skin. How do they penetrate the skin? Microorganisms can enter a little scratch that you may have in your skin, and maybe you're doing some gardening, you have a little cut. They can also penetrate your skin via a vector, a mosquito bite, a flea bite, yes, a tick bite. That's right, those are called zoonotic infections, and we'll talk more about that after exam 3. They can also enter through hair follicles and sweat glands, right? They can go into the sweat gland, they can go in through the sweat glands, and then there's a lot of nutrients once they get and penetrate your skin. So that is one way, penetration of the skin. Another way is that pathogens can penetrate the mucous membrane. Ooh, mucous membrane. All right, so that would be uptake by epithelial cells and antigen-presenting cells. So let's take a look at what do I mean by mucous membrane. Well, the mucous membrane is going to consist of the respiratory tract and the gastrointestinal tract. This is a great table here that you do not need to memorize it, but if you see the number of organisms and the diseases associated with those microorganisms that you usually contract through the respiratory tract, right? Look at all this. And here you see all the microorganisms and the diseases associated with those microorganisms that enter your system through the gastrointestinal tract. Now, most of these organisms and diseases we're actually going to talk about for the final exam material. Actually, like 90% of these organisms we're going to talk about. The mucous membrane also includes the genitourinary tract. So a lot of these would be your sexually transmitted diseases or STD. So you can see that there's a lot of different pathogens that actually enter through the mucous membrane, whether it's a respiratory tract, gastrointestinal tract, or genitourinary tract. The other portal of entry that we talked about is the skin, right, or parental route. And so through here, we see these various organisms and the diseases associated with them. And I didn't mean to show you the answer here, but which is the more common route of infection? Is it the skin or the mucous membrane? And just looking at this extensive list, right, you see that it is the mucous membrane, which includes the gastrointestinal and the respiratory tract to be the most within that group. Okay? Okay. Let me just go back to this. So most of the microorganisms are going to enter through the mucous membrane of the gastrointestinal tract and the respiratory tract. Why? Why is that? Well, when you think about that, the respiratory tract is the easiest, right? And the most frequently traveled portal of entry for infectious microorganisms. How so? Well, microorganisms are inhaled to the nose or through the mouth. When you think about someone having a cold, tuberculosis, flu, measles, smallpox, now COVID-19, right? Coronavirus. How do you contract it? Through inhalation, right? Okay. All right, so let's talk about this. Establishment of infection. Pathogens, once they enter, they're going to adhere at their portal of entry. And the way that they do this is via a ligand. So there's a surface molecule. You see, this is going to be specific. Adherence of the pathogen is going to allow the microorganism to bind to specific cell types. It's not just random. There actually has to be a connection, right? Once we see that the receptor binds to the ligand of the pathogen can go in. Infection occurs. There is replication of the microorganism. And now that the microorganism is thriving in its new environment, it can go on and proceed to damage the host. And the way that it damages the host is by a variety of mechanisms. It maybe exerts its toxins that it produces. It can invade the tissue, right? Various different ways. So how are microorganisms able to evade the host defenses? Remember all the different host defenses that we've talked about. Well, they're able to hide within the host. They can evade complement. You remember complement? We talked about the complement system. They can evade phagocytosis. Oh, I wonder how they can evade phagocytosis. They can avoid antibodies. And they're able to produce exoenzymes, as well as have some sort of antigenic variation. So these are all the different ways that microorganisms can evade our host defenses. So this is a very nice slide that sort of shows you the various different ways that microorganisms can avoid destruction by the phagocyte. We went over this slide before when we were talking about how phagocytes can destroy pathogens, right? Usually what happens is here we see a pathogen, a microorganism. It's going to be opsonized, right? C3b will coat this pathogen, and it sort of serves as a marker to increase phagocytosis. So this phagocyte is going to come in, it's going to engulf it, which is what naturally usually happens. And this antigen or this microorganism is going to be enclosed in a phagosome. This phagosome is going to fuse with a lysosome. Here's the lysosome. And when it fuses with the lysosome, now the killing is occurring. This process usually takes about 20 minutes, right? And now that the microorganism has been broken down, it will be released. Well, guess what? There are microorganisms that are able to evade every single step that we just outlined. In fact, there are microorganisms that will prevent this initial step, prevent the encounter with the phagocyte. Can you think of a way that pathogens can evade even being phagocytosed? Capsule, that's right. And there are microorganisms that will evade being in this phagolysosome or phagosome. So there are various different ways that microorganisms can avoid every step of the way. Okay. Here is a microorganism, and this microorganism is a diplococci, and you can see the capsule that surrounds this microorganism. This is one of the stains that you performed in the lab before we left. Okay. And so what I want to do is I want to show you one of the ways that microorganisms actually evade phagocytosis. Okay, so let's take a look. All right, here we go. Now remember that Staphylococcus epidermidis, we've talked a lot about Staphylococcus epidermidis, is normal flora of the skin, right? So here we go, Staphylococcus epidermidis. We need this normal flora in our skin, right? It protects us against more pathogenic microorganisms. So it's good to have. Now we have its close cousin on the other side, Staphylococcus aureus. Notorious for causing different types of infections, skin infections, even gastrointestinal infections, but we'll talk about those later, right? How is it that Staphylococcus aureus can cause infections and Staphylococcus epidermidis doesn't? Now remember that every microorganism, given the opportunity, can cause disease. Okay, so here we go. Here is my Staphylococcus epidermidis. It's a cocci, right? And it's going to have these little epitopes. So let's say that it got into an area that it doesn't belong. Well, not to worry, because my little antibody is going to recognize this as, hey, you don't belong here, right? Here's my antibody. And what does the antibody, where does the antibody bind to the phagocyte? Here's my phagocyte. This is my phagocyte. Right? How does the antibody bind to the phagocyte? Do you remember? It's going to bind, the phagocyte's going to bind to the Fc portion of the antibody, correct? Okay. So this is my S-epi. This is how, if this microorganism happens to get into a location that it doesn't belong, let's say it gets into your bloodstream, for example, or you have a cut for whatever reason, this is how Staphylococcus epidermidis would get eliminated. Yeah? Okay. Now let's take a look at its close cousin, Staphylococcus aureus. Staphylococcus aureus, also a cocci. It has its epitopes. But guess what else it has? It has a special different protein here. Okay? And this is called protein A. This is what I would refer to as a virulence factor for the organism. How is this a virulence factor? Well, guess what protein A does? What protein A will do is it basically won't allow the antibody to bind to the epitopes because it's going to intercept essentially and bind to the Fc portion of the antibody. Is that a problem? Here's my phagocyte. Is that a problem? What do you think? Can this antibody bring this bacteria to the phagocyte? No, it's not going to happen. It's not going to happen, right? Because remember that this phagocyte binds to the Fc portion. Therefore, it's not going to happen. All right, let's continue. Now that we outlined it on the whiteboard, let's go ahead and take a look at what this looks like on the actual slide. So here we have are avoiding opsonizing antibodies, protein A. Surface protein that is found in the cell wall of Staphylococcus aureus is going to bind IgG at the FC region. This is going to result in the bacteria binding the IgG in the wrong orientation, which is going to disrupt opsonization. It disrupts opsonization and phagocytosis. See, Staphylococcus aureus, it has its epitopes, but in addition, it has this protein A that binds the antibody in the wrong orientation, therefore disrupting opsonization and phagocytosis. Whereas here on the left, we see our little friend Staphylococcus epidermidis. It has its epitopes. The antibody will work as it was intended to. Here's my IgG antibody. It's going to bind the epitope the way that it should. And it brings it back to the phagocyte where it will bind to the phagocyte at the FC portion of the antibody. Continuing on, exoenzymes. These are going to be extracellular enzymes that are produced by the microorganism and then released into its environment. These include coagulase, which can clot fibrin in the blood. It can include kinases, which will digest clots, and collagenase, which breaks down collagen. Exoenzymes. These are soluble proteins. Soluble proteins produced by gram-positive and gram-negative organisms, but they're mostly produced by gram-positive organisms. These are similar to endotoxins, okay? And there's a variety of them that are produced. And we'll talk about the diphtheria toxin soon. Microorganisms damaged by host cells. Okay, so how? The mechanism of the pathogenicity. They can use host nutrients. That's how they can damage the cell. They can cause direct damage by invasion, their sheer numbers. They can produce toxins like the ones that I just mentioned to you. We're going to get into the toxins in just a little bit, the specifics. They can induce hypersensitivity reactions as well, right? So we're going to focus on the toxin productions, and then I'll talk to you a little bit about these hypersensitivity reaction as it pertains to toxic shock syndrome. So this is one of the ways that microorganisms can damage host cells. They can produce toxins. Two types of toxins that microorganisms can produce. Toxins can be exotoxins. These are going to be secreted by living bacteria, produced inside of the cell, and secreted out into its environment. Exotoxins. Diphtheria toxin is one of the toxins that's produced by a microorganism called Corynebacterium diphtheriae. And this is a very virulent pathogen. It causes the respiratory system to form this thick pseudomembrane in the back of the throat, and it causes suffocation. Very, very, very virulent. One of the interesting things about the diphtheria toxin is that it only produces it when that bacteria is actually infected by a lysogenic phage carrying toxicity gene. Wow, what does that mean? Think about what that means. Okay? Most of these exotoxins can be destroyed by heating, but not all. Right? So we'll talk about that later when we talk about diseases. The other type of toxins that microorganisms can produce or have are endotoxins, and usually these are going to be natural compounds that are found inside of the bacteria, and they're released mainly when the bacteria is lysed. So LPS, we've talked about LPS. Usually for gram-negative organisms, it's going to be derived from LPS. Endotoxins are usually from gram-negative microorganisms. There is, however, one gram-positive organism that produces an endotoxin-like toxin, and that is Listeria monocytogenes. It's endotoxin-like. This is a very bad pathogen to have. It causes meningitis in newborns. and it's usually acquired transvaginally. This is one of the reasons why pregnant moms are advised not to eat soft cheeses such as brie, camembert, queso fresco, things like that. And usually, and even deli meats, really, because this Listeria monocytogenes is a microorganism that can actually grow at refrigerated temperatures. And it is notorious for contaminating deli meats and soft cheeses. If you are a healthy individual and you eat something that's contaminated with Listeria monocytogenes, it's not really going to cause life-threatening injury. Okay, even if the mom eats it, she may not even realize it, but it's the baby that can get affected. And so that's one of the reasons that you should try to avoid that when you're pregnant. All right, so let's continue on with this exotoxin. Here is an example of exotoxins on the left. They're produced inside of the cell and then released to their environment, and they can affect various organs, as you see here, right? As opposed to the right side, these are endotoxins that are produced inside of the cell, and they are released when the microorganism is lysed, and these usually result in fever. These are the various different categories of exotoxins. And we're going to go over the top three. Okay? Categorize toxin by mechanism. The first one is an AB toxin. It consists of two parts, the A and the B. Most exotoxins are going to be AB toxins. Second is membrane damaging toxins. They cause lysis of the host cell by disrupting their plasma membrane. And the third are your super antigens. These are antigens that are going to provoke an intense immune response. They non-specifically stimulate proliferation of immune cells that are called T cells. The LD50 is one of the ways that it's the way that we measure and compare the potency of different toxins. Okay, so LD50 stands for lethal dose for 50% exposed. How much toxin does it take to kill 50% of the population? Okay, Clostridium botulinum prevents transmission of nerve muscle stimuli, and we know it's been calculated, extrapolated data, right? That 1 milligram of botulinum toxin is so toxic that it's enough to kill 1 million guinea pigs. Did they really kill 1 million guinea pigs? Hopefully not. It's extrapolated data. This is a nice table. It's not from your book, but I love this table because it shows you the various microorganisms that produce exotoxins, the diseases that they cause, and the type of exotoxin that is produced. And as you can see, the majority of them do produce an AB toxin. So let's go ahead and outline this AB exotoxin. AB exotoxin has two protein parts, okay, the A and the B. The A is going to be your active enzyme component. It has a toxin function. The B is going to be the binding component. Okay? So here we see that in the first part, we have the AB toxin is released from the bacterium. The B part is going to bind the surface receptor on a cell. Usually it's a carbohydrate that it binds to. Following the binding, the AB toxin is going to be transported across the plasma membrane into the host cell cytoplasm. Now within the cell, as you see here, within the cell, the AB component separate. A is going to go ahead and it's going to inhibit protein synthesis and kill the host cell, whereas B will go on to be released from the cell. Other types of let's continue on with the different types of AB toxins. There are neurotoxins that damage the nervous system. An example of that is botulism. Botulism is an AB toxin. Yeah. Enterotoxins cause intestinal symptoms such as the cholera toxin. We're going to talk about cholera when we talk about diseases, and one of the terrible effects or side effects of a cholera intoxication has to do with the toxin that the cholera produces. Cytotoxins as well, they lyse or interfere with cell function, diphtheria toxin. But some of these toxins that are AB toxins have some commercial value. Specifically is going to be the Botox, right? Botox, which is the instant facelift. So with the Botox injection, they actually are injecting the A part of the toxin. Yes, so it is toxic. It is. You're injecting poison, right? And I mean, it has clear effects, right? Really good effects, but it does take upkeep. Usually it lasts about 3 months, so there's upkeep to be considered with Botox. All right, let's move on to the other types of toxins or exotoxins. Is going to be membrane damaging toxins. These are going to disrupt the plasma membrane and lead to host cell lysis. Now, there are 2 different types of membrane damaging toxins, the pore formation and phospholipases. Okay? Pore formation and phospholipases. So these are cause lysis of the host cell by disrupting their plasma membrane. They can do this by pore formation, in which protein channels are actually formed in the plasma membrane, or by phospholipases, which disrupt the phospholipid portion of the membrane. This is an example of pore formation. Whoa, that looks like lysis, hemolysis, right? That's exactly what it is. There's, this is pore formation. There's also membrane can disrupt toxins that destroy red blood cells by forming these protein channels. This is referred to as hemolysins. Streptococci and staphylococci can actually produce this, and you'll see this. For phospholipases, here we see the normal cell membrane with our lipid bilayer, right? And if the organism releases phospholipase exotoxin, you're going to see that it's going to disrupt this phospholipid bilayer. Gram-positive rod, a gram-positive rod that causes gas gangrene is an example of an organism. It's called Clostridium tetani, it's a Clostridium. It's going to actually produce this exotoxin phospholipase, and it disrupts the phospholipid. And that contributes, as you can imagine, to the virulence of the organism by killing the host. Yes, it kills a host. The third type of exotoxin is that of superantigens. Superantigens are so called because of their ability to activate a large number of T cells. Okay, a large number of T cells. The disease is going to be characterized by fever and shock. Superantigens are toxins that are produced by Staphylococcus aureus as well as Streptococcus pyogenes, and they possess a very potent immunostimulatory properties and the ability to cross-link. So that's going to be key. The ability to cross-link what? MHC2 that you see below here with the T cell receptor. You see in the middle that it has the antigen, right? So what forms is what's referred to as a trimolecular complex. Trimolecular complex of the MHC2, the T cell receptor or TCR, with the superantigen. There it is. You see that? There is a superantigen. This trimolecular complex, and here's the antigen in the middle, look, the little antigen that provoked this interaction. This trimolecular complex of the MHC2, superantigen, and TCR induces the proliferation and cytokine production in T cells. But to such levels, because remember that it activates a large fraction of T cells, to such levels that it actually results in toxic shock. There are some food poisoning symptoms that can result from this, as well as scarlet fever. So it is a very intense reaction, non-specific mass activation of T cells that causes this toxic shock. Endotoxin and endotoxic endotoxin and endotoxic shock. Endotoxin is we usually refer to the LPS, the lipopolysaccharide that is found in gram-negative organisms. We know that the active portion is the lipid A. The source of the endotoxin, we know that it's in the outer membrane of gram-negative organisms. How does it get released? Lysis. Lysis that occurs of the bacterial cell by various means. It just dies. It is killed by the immune system. Maybe it's killed by the antibiotics that you are taking, right? Just a quick little review. Where is the LPS? I don't remember. That was a long time ago. So remember that the LPS is on the outside here, on the outer membrane. We see that the lipid A is embedded in the plasma membrane, and the O polysaccharide is what sticks out. Endotoxin can stimulate fever. How so? Well, let's take a look. Here we see that the gram-negative bacterium is ingested by a phagocyte, just the way it's supposed to work. Now, when the bacteria is degraded, you see that it's being degraded, it's going to release its LPS. It's endotoxin. That's going to cause macrophages to produce interleukin-1 or IL-1, which is a cytokine. The cytokines are going to travel to the hypothalamus of the brain, right here, the hypothalamus, which is the site that controls temperature. Yeah. As a result, there's going to be a release of prostaglandins that results in fever. Whoa, it results in fever. What are the effects of endotoxin? Well, it can damage, there can be damage associated with inflammation. LPS is going to activate both the innate and adaptive immune response. It does, right? Macrophages are going to release these cytokines. Fever induces inflammation. B cells can be non-specifically activated, right? Now, here's something to think about. Antibiotic treatment can worsen the effects of endotoxin. And antibiotic treatment can worsen the effects of endotoxin. How so? Think about that question. Think about that. We know that antibiotics used to treat gram-negative bacteria, well, what do antibiotics do? They kill the bacteria, right? So if the antibiotic that you're using to treat your gram-negative infection kills the bacteria, what happens when the gram-negative bacteria is killed? As it's lysed, it releases the endotoxin, right? And so that can result in an immediate worsening of symptoms. So sometimes we refer to antibiotics when treating gram-negative infections as a double-edged sword. Patient's already sick. It's very possible, it's not impossible, or it's very likely that the patient may actually get worse before they start getting better, right? Of course, the condition improves as the endotoxin breaks down. But it's not unusual that the patient may actually get worse with the treatment of antibiotics as the bacteria is being killed and its endotoxin release before they get better, okay? So we know that endotoxin stimulates macrophages to release cytokines in high concentrations. Can cause fever, weakness, aches, shock, and even death. So septic shock, endotoxin in large amounts is going to trigger systemic inflammation and blood clotting proteins. DIC stands for disseminated intravascular clotting. And this is actually severe coagulation that can be fatal. And it's due to endotoxin activating blood clotting proteins that cause the formation of small blood clots. Okay. This is some great tables. They're not again in your book, but I like to include them here. They show you the basic generalities of exotoxins versus endotoxins. Just gives you a little bit more information. And now we get to the end here. Portal of exit for microorganisms is going to allow for the spread of infection. Bodily secretions, this is how you can secrete these microorganisms and spread them. Remember that the portal of exit is usually the same as the portal of entry. How you got infected is the way that you're going to disseminate this microorganism and possibly infect others. And this could be through bodily secretions, whether it's mucus, whether sneezing and coughing, feces, vaginal and semen, for STD specifically, blood, skin shedding, right? Yeah. And so that concludes today's lecture.